Oh, okay, cool. Okay, thanks. Yeah, so back to our 50s, 60s, and 90s story. Okay, so now now we are in the 90s, okay, and the idea of looking at the whole genome and sequencing the whole genetic variation, the SNPs, and everything in the whole genome comes to the fore. So how has this been done in terms of for pain? So, oh, excuse me. Oh, okay. So we're in the 90s, and around that same time, the American Pain Society, that was in 1995, kind of met together and then began to think about, oh, can we begin to include pain as a fifth vital sign, right? So there was that campaign and came up with a numerical pain rating scale with the goal of using that to standardize and study pain. And in 1999, that was about 4 years later, the VA included the pain NRS as part of what veterans are often asked. So they often ask, are you in pain? Can you rate your pain on a scale of 0 to 10? And then it was adopted. The goal of, again, using it to track pain severity over time. But as much as that worked, there was a ripple effect. And what was that? There was a rise in prescription opioid use. Okay. And then opioid overdose deaths in the 2000s. There was wave one, there was wave two, which then there was a rise in heroin overdose deaths. That was wave two, that's 2010, right? So if you remember where we're coming from, we can look at the whole genome to see if there is anything related to pain. That's for GWAS, okay? Now, wave three, a lot more is going on. So 2013, there was a serious rise in synthetic opioid or overdose deaths, meaning opioids, as much as they work for some certain types of pain, they're not working, okay? So we need to look elsewhere, which is why it's important to do GWASs of pain. I don't know if that makes any sense. So with that in mind, some studies apparently, but not in the US. So which is now the issue. Now, some studies using genetic data from the UK Biobank, which is in the UK, by the way, looked at regional pain conditions. Pain in the back, pain maybe in pain, I think neck pain, back pain, knee pain, they looked at these different conditions and looked at the whole genome with the goal of identifying genetic variants, and they found a few, okay? But the issue is most of the genetic variants they found did not overlap across the different pain conditions. So that was one issue. The second issue was still around that time, much of the studies were on individuals of European ancestry. All right. So which then bears to fall the most recorded pain issue we know is in the US. And what has been done in terms of looking at the whole genome and identifying the genetic variants for pain in the US? Almost zero. All right. So, which is why we then came in and we're like, okay, you know what? We need to do something. Okay. And what do we need to do? We need to in order to kind of develop new therapeutic targets for pain, we need to open it to everybody. Okay. And in that sense, we look at the etiology of pain. We look at the underlying biology of the genetic variation. We want to be sure that those who are at high risk for pain or individuals who experience serious side effects from these drugs, we are taking care of them as well. The other issue is we want to identify those who are at high risk and then look for the right intervention. Okay. Now, to do that, we started with a GWAS of pain intensity, pain severity. And this is like way after COVID. So if you look at it properly, you see that we had a lot of lag in time to do a GWAS of pain. For a number of years, it hadn't been done within the US context. So we took data from the VA. We looked at the pain numerical rating scale, and then did a GWAS of the pain scores we found in the VA, a lot of them, millions of pain scores. And we found about 125 genetic loci, like locations in the gene that were associated with pain intensity. 66 of these have not been related to pain in any study prior to this time, so which was really good, right? And this was cross ancestry. The other thing we found was the genetic correlation for pain, which is like how much of the genetics of pain is shared with other conditions. It's really huge. For pain intensity and other pain conditions, you see there in green, there's a high correlation of pain intensity with other pain conditions, but not that alone. If you look right in the middle, you would see the psychiatric conditions, which is in blue. For example, you would see ADHD, MDD, major depression. You see, I think there is also cannabis use disorder. There's opioid use disorder. So what we showed was that as much as we want to look at the genetics of pain, it's important to look at the genetics of pain with other conditions. That's stage one. Stage two, not just in individuals of European ancestry, but in individuals of African ancestry. So we showed that this shared genetics is not just limited to people of certain ancestry groups. It's sparse across everybody. Okay, so, and it was well received. And the other thing we found was that the underlying biology that explains this genetic risk is largely in the brain. Okay, so I'll just put that out there. So in the plot A, you see that what we are showing is how much of the genetics of that pain intensity is enriched in different tissues of the body. Okay, and you see the dotted line there showing that with the pink CNS, which is the central nervous system, is highly enriched, more enriched compared to other parts, other tissues in the body, which is interesting. And the other thing we did was, what are the genes that this genetic loci, or I'll call them single nucleotide polymorphism, these simple tweaks in the genome, which of the genes are they related to? So we found a few, and we found that in panel D, some of them could be going up, and some of them could be coming down in relation to pain. So which is really good in terms of the dynamics of pain. The other thing we found was beyond just the tissue in the brain, can we identify specific cells, right? So if you remember your high school biology, you go from your cell to your tissue, to your organ, and to your system, right? So if we take that back all the way, so you look at the system, the organ, we are showing that it's in the brain, in the tissue. Now, if we come to the level of the cell, what we showed here is that the GABAergic neurons, so these are like inhibitory neurons, right, are actually well involved in terms of pain intensity. So this was well received, and we got a few press attention and all that, but that was not all. The other thing we also found was when we take these individual genes, we found, and we integrate them with current drug use, or current drugs, I would say, that have been used to treat different conditions, what we find is an interaction of some of these genes with multiple drugs. So I'll take just one, for example. So if you look at the GRK4, which is, I think, a glucose kinase receptor 4, that gene has been related. We show that that gene actually relates to drugs that are currently being used to treat, I think they are beta blockers, okay? Now, those drugs are not currently being used to treat pain, but they are being used to treat other conditions. So that opens up repurposing, I would say, opportunities for that particular gene to be used as a target. So there is a lot in that. If you're interested, the paper is currently published last year. So I'll just, for the sake of time, I'll move on. Now, the other thing we're interested in is, now we've done this in the MVP, okay? beyond the MVP, what else can we see? And why did we think it that way? It's because number one, the MVP is 90% male. Okay. Number two, it's not I wouldn't say it's not a it doesn't give a global representation of what's going on. So it's just veterans. So this could be, of course, we know them to be really high PTSD, high depression, all right? But we could integrate these with other data sets that have good balance in terms of male and female. So we did that. We combined our data with other biobanks in Europe and increased the sample size to 1.2 million. And we found more, okay? So which was great. One of the things we found was that the genetic liability for pain is not
Sorry to
just
interrupt you.
shared, but the
Hello?
genetic variants that are involved in pain are actually shared with other conditions. So for example, here I'm showing you there is a high
Sylvanus,
proportion of
your
genetic
slides don't
variants that are
show.
shared with insomnia for pain, high for neuroticism. So these are like psychiatric related conditions.
Your
High for
slides
depression,
don't
which is what we
show.
expect, for PTSD, for substance use disorders, so which is again what we expect, but this hasn't been shown before. For opioid use disorder, right? And in a very interesting way, we also found some level of shared genetic variation for the C-reactive protein. That's an inflammatory mark. So it doesn't just, we are not only saying that pain is brain, okay, but we are saying that there is an immune component to pain. And in terms of the genetics, we are seeing that effect here. So we then were interested in what's the direction of effect. Okay. So in that plot I'm showing, panel B is where I want to put our attention on. You can see in panel B towards your right, some dots in red. So what we did here is to look at brain structures. These are IDPs. These are inferred brain measurements. And what we found was that total surface area in the left hemisphere and total surface area in the right hemisphere, a reduced total surface area actually is related to chronic pain liability, which again has not been shown before. And we were excited about that. We also showed that there is an influence of chronic pain on C-reactive protein level. We're excited about that. We're like, okay, fine, good. Let's go to the next thing. Okay. And here, this came up as a result of the previous paper. So when we were doing that study, we thought if we want to look at the pleiotropy of pain, it's also important to look at what specific cell types, not just in humans, but in non-humans. So this is what we did. And we integrated that data with brain data, with dorsal root ganglia data, which is the peripheral, I would say, neuron that often gets triggered when a person feels pain and then sends a signal to the brain. So we found enrichment not just in the brain, but we also found enrichment in dorsal root ganglia, both in humans and in mice. And much of the pathways we see are related to how a person processes their pain, either peripheral or in the brain. So I'll quickly want to summarize. So firstly, I'd say pain severity cuts across individuals of all ancestry backgrounds. We've been able to show using the Million Veteran Program data to that the genetics of pain intensity is there in individuals of European and African ancestry as well as Hispanics. We think that the genetics is of course shared with other conditions, so it's not just pain on its own. We have been able to increase, I would say, the number of genes that have been associated with pain. We've pointed attention to a few targets. And we are now saying that it's important to look at both excitatory and inhibitory neurons if you will want to develop drug targets for pain. And hopefully that would open up new opportunities going forward. And I would like to round up by thanking everyone. Thank you for sitting around listening to me, and also everyone who has contributed to making this a success. You can see the lovely faces in the picture right there. And then funding from all these agencies, the biobank data, and these individuals with whom I've collaborated down the line. And yes, I'll take questions. Thank you.
Genetic reason that people develop depression and are like are there with like are we able to figure out what's first or the depression or the pain can genetics sort of help with that?
Yeah, that's a good question. So one of the things we found is it's bidirectional. In terms of the genetics, is bidirectional for pain and psychiatric conditions. But in terms of the neurobiology, we are seeing similar pathways, but we are not sure yet. We, uh, I would guess there would be. So, for example, studies that looked at the genetics of psychiatric conditions also found the enrichment of, for example, glutamatergic neurons for depression. Now we are finding it also for pain. So that could suggest common pathways, but we would need further studies to establish that. Now, number three, I would say to be able to disentangle pain from psychiatry, that would be complex. But there are tools that can help to do that. So I'm not sure if anyone mentioned that, but there is a method called GWAS by subtraction. So basically, what that does is you put in a bunch of genetic studies of say pain, psychiatric conditions, okay, and then you then ask the question, how much of the common heredity or common liability can we see when we put them together, and then when we remove one, is there any difference between that? Well, I would say in terms of thinking about that methodologically, well, it can work, but I'm not certain if that solves the issue. My current thinking is we need to have the individuals to be able to do the study. So, for example, in the MVP, for example, we can do a pain psychiatric comorbidity type of study. I think Emily did some for AUD and depression, I think. So we could do something in that realm as well. Look at pain and other psychiatric conditions and see, okay, we have the controls who don't have pain and who don't have any of the psychiatric conditions, right? And then we have a group that has just pain, and then we have a group that has both pain and psychiatric conditions, and then test to see if there's any difference in the genetics. We could do that. I haven't even thought about that. Just anyway, but we could do that. That's doable. Maybe that would help us explain some of these commonalities, but it hasn't been done yet.
Do people study pain in young or since so it could be a lot of people, adolescents obviously develop mental health concerns and psychiatric issues, but I wonder if you like and even like how people over time, and does the genetics check like selecting like before they develop a psychiatric disorder that have but you maybe have some kind of pain disorder to people do that or I guess can I guess like how genes change these every time or something.
Yeah, that's a very clever, yeah, clever question. Yeah, I mean, it's
I know you're
actually
dealing
one
with
of the
really large data sets, right? So like having like a longitudinal data set maybe like that.
yeah, that sounds like M3, Mike. So in essence, what we can do for that is there is this cohort called the ABCD cohort. So we could do this, we could do a GWAS and then test to see if there's any association with any of the traits in that cohort. Oh yeah. Yes, so it's adolescent brain cognitive, what's the D? Yes. So in essence, that study kind of recruits adolescents to see if there's any pre-existing condition that is related to the genetics. And that, yeah, yeah, yeah. And they also, I think they also have imaging data as well. So yeah, that's a good resource to test the hypothesis you mentioned. Um, I mean, that is if there is any record related to pain phenotypically in that cohort. Because if there isn't, right? But there, I know there is for substance use because we've used that study for a recent GWAS we did for tobacco use disorder. And we found some association with certain substance use behaviors in that cohort. So I don't know if they have pain questionnaires. That would be worth looking at.
So
Yeah.
you have a long
Um, it's suggested, but and actually I think Crystal is now beginning to dig into some pain trajectory type of analysis. And hopefully we'll get something from it. So the idea there um, is there a way to look at the development of pain over time? And the way the VA data was set up allows us to answer that question because the pain scores, the pain NRS, as is there in the biobank, is there in the corporate warehouse, what do they call CDW, corporate data warehouse, for about 18 years worth of pain scores. So there is that longitudinal framework to it that allows us to be able to test if maybe the increase individuals who fall into the trajectory of increased pain over time, is there any genetics behind it compared to those who are like normal over just moderate over time, or those who are like whose pain scores fall over time. So there are ways to look at that, and we are currently digging into that. I think Crystal is doing the phenotyping of that now, and we'll see. We might find something. Hoping we do. If we don't, we'll see. But again, I think we might because we have the numbers. This is like almost 600,000 individuals, right? And currently we have more than 100 million pain scores. So I think in terms of numbers, we might be good, but again, science, yeah, might say no, say yay, your idea was not, yeah, but we'll see. But keep your fingers crossed. I think there will be something coming up shortly on that.
I'm curious about maybe sort of like a recruitment and classification question. We're talking about like people of African and European and Hispanic ancestry, where the borders are drawn on that, right? Like, well, first of all, I suppose whenever recruiting people, we just ask them how do you identify yourself as like white, or is it like what's your actual ancestry from? Because if you go by like categories of whiteness, then that would include like Southwest Asians, like Armenians, even Iranians, people from Iran. And then so that's like one sort of factor that I'm thinking about. But then also, especially with Hispanic populations, I mean, that is sort of inherently a multi-ethnic thing of like European, African, and indigenous ancestry. So I'm just wondering how that could be like potentially confounding if you don't have like really strict regulations on where you draw those boundaries.
Yeah, that's another brilliant question, I must say. So, I would say there are like two ways to address that. Number one, initially in the MVP, Million Veteran Program, right? Ancestry was defined using hair. I've forgotten what hair is. It's some form of, I think it's more of clinical way of saying, oh, okay, only identify us, right? But now there is genetically inferred ancestry, such that we infer the ancestry of these individuals irrespective of what they ticked in the box using their genetic data. I don't know if that, so that gets at um, subtle differences that may occur. So that's one way to look at it. The other way to look at it is we now have tools that allow us to do like trans ancestry type of genetic studies, such that we don't care whatever ancestry is being recorded. We just put everything in one bit, in one chunk, and then do the GWAS with that. So there's one tool called Tractor that could allow one to do that. Then the use of, for example, the genetically inferred ancestry would help us get at more of the biological questions. I don't know if that makes any sense. So in that way, by saying, and sometimes what we found is there might still be some issues in that, but we think it's close enough. Rather, let me put it this way. We think it's farther enough from the issue of classifying ancestry based on just self-report. I don't know if that makes any sense.
Yeah, yeah.
Yeah.
Like, I think for me, one of the big difficulties, or I suppose just a big difficulty in there with like ancestry type stuff is we on a policy level like to treat race as a genetic thing, but really it's like a sociogenetic thing. It's like a lot of the boundaries that we create are just purely cultural. So I suppose that explains how you like, get bypass that type of problem.
Yeah. I mean, yeah. And I know it's one of those sensitive things. I remember there was this paper that was published, was it last year, that used data from the All of Us cohort. That cohort, again, it's called All of Us. It's here in the US with the intention, they recruited individuals of different ancestry. It's really diverse, all right? And then they plotted ancestry using some multi-dimensional way of looking at it. And the people pick issues with it and say, hey, why did you use that and all that? Again, for example, one thing that was recently established in terms of, let me use this, polygenic scores, is that it could be a continuum. It's not just you are this, you are that. It could be like a continuum of differences across a spectrum. Okay, so there are different ways to look at it, and people are beginning to link genetics to it, and it's an area of serious interest. You probably might want to even speak with Kira on that. She might have a lot to say because she's part of the Psych Image Diversity Network, and that's something of serious interest. Yeah, but for us, for example, what we've done mostly is we have now we have genetically inferred ancestry, and we run our genetic analysis using that inferred ancestry from the genetic data. And that way, we just call it Okay, these are individuals of African ancestry based on their genetic data. We write it up that way. So
Are you expecting, as you are kind of able to look at different types of pain, some of those like drug repurposing and enrichment results to like shift? Because I'm just kind of, I know we kind of lumped everyone in together for the first paper, and now you're looking at chronic pain. I'm thinking about like fibromyalgia is really different than like, oh, I broke a leg and I got hooked on weights. So you know what I mean?
that's complex, I must say. Now the reason why I say it's complex is okay. For example, in our most recent paper that dig a little bit into the cell type analysis to see if a particular pain condition is related to one cell type or is there common cell types across the different pain conditions. We don't have the numbers yet. So why do I say that? So for example, the genetic study for back pain, the sample size is like this way. But the genetic study, for example, with fibromyalgia, the sample size is like this way. So we can't tell if any of the enrichment we are seeing is driven by sample size.
Yeah.
So until we are able to control for that difference, I think for me, that's me speaking, I would then be able to confidently say, okay, I think that could be the cell type. But right now, a lot of the enrichment we are seeing is powerfully driven by sample size, I think. That's one. But it doesn't mean the biology isn't there. So for example, in the paper, we found that glutamatergic neurons, especially in the interneurons, so these are like neurons within neurons, okay, are related to pain. When we look at peripheral neurons, we find enrichment in C fibers. Okay, so these are like neurons that transfer, I think, slow pain, slow dull pain. Okay, so you then begin to feel, okay, well, slow dull pain, I would feel that more in this part of my body, and I will feel a sharp pain in this part of my body more. I don't know if that makes any sense. So if I'm looking at leg pain, well, if I take the DRG, the dorsal root ganglia, well, would I expect to see a sharp pain or a dull pain? And it relates also, it becomes more complex. So again, would we say someone who got hit by a bus, would the pain be dull or sharp? Which neurons would transfer that type of pain? How many of such persons can we get for a genetic study? How many cases do we need? How many controls do we need? So it's as much as it's in real time, it's very interesting to do. I think we're a bit short in terms of sample size. But right now, if you ask me point blank and say, for the brain, which neurons should I look at? I would tell you, look at glutamatergic neurons, right? For chronic pain generally. But if you are interested in specific types of pain, say, for example, you want to look at the severity of the pain, then I would say, well, don't just look at glutamatergic neurons, look at GABAergic neurons. Those are like the inhibitory neurons, because it could be a combination of both, really creating that process of you being able to perceive your pain, you being able to transfer relay the information about how your pain feels and if it's high or low and all that. So I think we need animal studies to be able to establish some of the things we found in humans. I think we need we also need some brain imaging studies to be able to confirm some of the things we found.